Until a few years ago, no one talked about organoids, and no one has ever thought of bringing organoids in the context of regenerative medicine.
This is really a change in the paradigm of cell culture.
I think that we are really getting closer and closer to a cellular therapy of diabetes type 1. LSFM4LIFE, it's an European project with the aim to translate organoids from the laboratory bench to the therapy of diabetes type 1.
So what are pancreas organoids? Generally speaking, organoids are three-dimensional cellular structure generated from induced pluripotent stem cells as well as from adult tissue, progenitor cells.
Well, I think the role of modern microscopy is actually that we are in a phase of transition where we move from monocultures or simple cultures, cells grown on flat and hard surfaces, to multicellular three-dimensional specimens.
Organoids are made by stem cells. They can differentiate into different types of cells and recreate the function of an organ. This is very different from cell lines or other cells which are used in the laboratory. These are really cells that have a potential to cure disease. In our lab, we are working a lot with microscopy, and we characterize how the organoid grow and how they differentiate by using light microscopy and specifically fluorescence microscopy.
Well, light sheet microscopy is very fast, first of all, and then of course what is very important is that the cells survive it. So whatever we look at, whether it's organoids or spheroids or also small model organisms, they survive very long periods of observation.
I'm sitting in the lab and doing organoid culture for expansion, for quality control reasons, and I do microscopy to obtain or generate numbers and parameters for the clinical application.
Now a part of LSFM4LIFE is to generate the human pancreas organoids, try to keep them in long-term expansion and differentiate them to beta-like cells.
My main involvement is testing the organoids in animal models in terms of their safety and how well they work.
Since the LSFM4LIFE project focuses on the development for us, on the development of a hydrogel for the culture of pancreatic cells, we needed to adjust our hydrogel system to the culture of these cells. And then we, of course, also set up culture conditions for making pancreatic organoids here.
First we started with the mouse organoids, so this was pretty easy. We found very quickly a gel that supported their growth and expansion. When we started with the human organoids, this was quite a different story. So it took us a while to find out which adhesion peptide, for example, would support their survival and their growth.
So our facility for more than 20 years has the role of translational and clinical production of cellular therapy. What does it mean, you would say? It means that we have to analyze all the reagents that are used in research setting and looking for suitable ones to be used in patients. Practically, it's a kind of a bridge activity between basic research and clinical application. And the second aspect of our work is the cellular therapies for clinical purposes. So cell therapies are under the GMP rules. GMP, what means GMP? GMP means good manufacturing practice. With the GMP we have to deal with the clean rooms, for example, with the control pressures, with particles and microbiological counts.
Working in a European consortium has the advantage that you can really bring expertise together that is otherwise not available locally. So a lot of the things that we do would not be possible in Frankfurt, but they are possible on a European scale. And that, of course, I think is really the whole issue.
Working in the European consortium, it's quite an exciting prospect because you're working with leaders in their various fields, and it's quite interesting to see how these different specialities, would it be chemistry with the hydrogels, biology on our part, imaging from Frankfurt, to see how they can really complement each other. And also you're in such a good position that you can come up with an idea and almost put it into practice straight away.
You have industry partners, you have academic partners, and you feel like you have all these kind of specialists in the fields and people that you can talk to about various parts of the project, but also that there's so many people in different fields that are working on the same kind of goal, so you really feel like you're part of a team and part of a group of people with the same goal, but adding different components to everything.
In the third year of the project, we are actually reached many of the objectives that we have set at the beginning. We can grow the organoids in a way which is safe for the patient, for a future transplantation. We are close to differentiate them to endocrine cells. I think that we are really getting closer and closer to a cellular therapy of diabetes type 1. The next step of LSFM4Life is bringing organoids to clinical trial.